And greetings, everyone. Dr. Mark Kelly. I probably just spent the last hour trying to get rid of my cameo on these slides, and I'm not sure how to do it. So hopefully I can go back into what they call post, which is called post editing, and shrink them or move them. But right now it's not cooperating with me. So yes, technology, and I'm not the best at it. I'm not the worst either, but I'm definitely not the best at it. So before these cameos, you used to have to insert them. Now it's you can't do anything to get rid of them. Well, we're going to get started into this first section. It's a lot of general stuff. It's a lot of introductory stuff and a lot of definitions. So I'll try to go slow enough, but I will emphasize the points that will be on the test. My tests are going to be coming from Test Bank, which is from the publisher. These slides are from the publisher. So everything should be pretty much cohesive, and we'll of course, go off and do a lot of other things in this course, but both the slides and tests, lecture tests, will come from the publisher's presentations. In this case, it's McGraw-Hill. All right, let's get started. Yeah. Um, there's various milestones in exercise physiology. I'm not going to go through them. I actually deleted some of the slides already on them, but it is quite fascinating. It's a relatively new science, and I remember meeting some of the just heavy hitters that started a lot of stuff back in the 80s at the ACSM meetings and some other meetings. So, but I've been in the field a long time. I've been a trainer, I've been a coach, of course, a teacher for the last couple of decades or so. All right. So as I was saying, one of the big differences between physiology and exercise physiology is that exercise physiology is a branch of physiology. Of course, it's a branch that's studying how different systems change during exercise. And it's exercise of all different kinds too. So they're showing two kind of sub areas of it, environmental factors. My dad was actually a aerospace physiologist, which they actually call themselves environmental physiologists, but it's specific different conditions. In his case, of course, space and even high altitude stuff. He did some studies in there, and then how the body responds in those situations. And I've also gotten into specific populations. I've done some stuff in the medical fitness area, so it's the use of physiology for medical purposes. And I did programs in obesity and in osteoporosis, but I'm kind of one in Alzheimer's as well. Anyway, one thing I also want to say is there's acute conditions, which is something happening to that person at that time, and then chronic, and that's usually what we refer to as training adaptation. So what happens uh, across time, basically, what they call longitudinal studies. So kind of keep those two definitions in mind. And as I mentioned, we will not be getting into these milestones. So we're going to keep going. There has been a lot of different publications across time, and of course, it's exploding in how fast they're increasing. Um, Also, note that there's a couple different ways you can look at exercise physiology: a reductionist approach and an integrative approach. The reductionist approach, and they have the kind of weird word here, reductionism, refers to study of a single molecule to better understand the intact system. Basically, they're trying to pull things apart to its essence and then study it in that essence, not in its natural surroundings. Um, yes, that in fact that's often called in vitro because they're really looking at it, as a cell, on a slide type thing, versus integrative physiology or systems physiology, which is definitely more what I'm about, is looking at things as a system. So the interactions between the different systems and then how they function. Like I said, and the study is looking at it as a single part almost. Um, anyway, those are two different things. We'll also look at some of these omics that are coming: genomics, transcriptomics, proteomics, metabolomics. So there's these interesting subcategories that are happening because of new developments in science and especially genetics. All right, and this is just showing you what the reductionist approach would do. But again, they're literally trying to look at contractile proteins. And of course, now we understand contractile proteins, and that's how muscles function. So both approaches are needed. It just depends on which one you prefer to use. And here's some, and I know this my cameo's disruptive right now of the letters in there, but genomics, of course, I think you guys know about that, the study of the structure function of all the genes collectively known as a genome. But transcriptomics, proteomics, of course, these lead to a protein which then affects our metabolism. So it's sort of the way that they're looking at different aspects of genes becoming proteins and then affecting our metabolism. Okay, looking at some of the what we call physiology research, the path knowledge. The scientific method is a set way to do things, and I'll go into some of the definitions of it or the details of it in a little bit. But we have different classes of research designs. There's field and laboratory research, of course, field being literally on the field in the playing field, in the end, and then basic and applied research. So these are different ways to be looking at it. And applied could be, how does someone hit a baseball better? You could actually apply that. Versus basic would be more, we'll call it foundational. It can be applied to a lot of different things. It's kind of basic here. So let's get into what is the research design. Hopefully I can use my pen here. Um, First of all, you ask a research question. Very often, though, you make some observation that leads to that question. Now, does a question have direction? No. Question is just that. Well, sure, you can have leading questions, as they say, or directive questions, but for the most part, questions don't have a direction. However, a hypothesis is a guess, basically. They used to always call it an educated guess. Well, what's the education? That you just saw something. You don't really have to be educated to have the guess, but it is a statement that you're saying it's this, not that. It's like placing a bet. You have to, I wonder who's going to win the game. No, you're saying I think the Rams are going to win the game, or Pittsburgh, or whatever it might be. The point is that you've made a statement in this hypothesis. Now, very often you're looking for some effect and change. So we'll call this a delta versus no change, and that's often known as the null hypothesis. So just kind of keep that in mind. And then you do an experiment to test the hypothesis. Then you collect and analyze data. Okay, so you collect, but how do you analyze data? Very often with statistics. So the statistics will tell you if your hypothesis was proven or not, or if it happened by random chance. Um, I I've even taught the course and I called it statistics because people hated it so much, but it was actually statistics. But again, you're interpreting the data based on those statistics, and then you formulate a conclusion, and then you communicate the results. Well, they often will call this conclusions and discussions. So this is the area, but this is where you're actually interpreting the data. So you're allowed to make an interpretation here. In the results, it's just the facts, man. So you are just showing the data, but you're not interpreting it. All right, so we'll keep going from here. So what is a non-experimental research? Done all over the place, very important stuff, and descriptive studies, case studies, and correlational studies. Now, we'll get into what a correlation is, but it's basically the effect of one variable on another but I just use the word effect. That's not the case. It's actually the relationship of one to another. We cannot say you have a cause and effect with a correlation. Literally, what it means is core, and then relation. So the two are correlated, or the effect. So there's often a term in this called the R value, which shows you the strength of that correlation. So those are done quite often. And then in experimental research, we will alter the independent variable, the IV, to then measure its effect on the DV. Now here, for the most part, we can claim a cause and effect relationship here. So what what effect did the independent variable have on the dependent variable? And a really good study that has a control group would have all the things the same except the independent variable was not manipulated for them. So that's the idea of a good control group, that you're controlling for all the different things that can affect it except that independent variable. Okay, so laboratory research, the advantages are it's isolated. Okay, so you can control these external things. The bad part is it's not realistic. Okay, so it allows invasive experimental protocols. You can control the environmental conditions, but it's not real world. Okay, so if I was running on a treadmill and the perfect conditions and versus running outside. Okay, so those are two different things. I'd have wind, I'd have uneven terrain, I'd have this, that, and the other. But if you were to run a race, where are you going to do it? On a treadmill? No, you're going to do it outside. So there's pluses and minuses to each. Okay, and again, basic research is trying to look at fundamental topics in biology. And of course, for us, it's physiology. Applied, though, is trying to solve a practical problem with it. And there's a kind of research called translational research, which tries to look at how to solve these things and evidence-based practice. So the translational research is actually a type of evidence-based practice. And if you ever heard of epistemology, sometimes they say that is basically looking at the evidence of something. And they're going into it a lot more in your book. So clinical applications on that. Go ahead and look at that on your own. Okay, so what is the scientific literature key points? Whenever you're looking at a true scientific journal, it's a peer reviewed journal. What does that mean? It means that to get published in that journal, somebody else looked at it that's also an expert in that area for its reliability, validity, if the facts made sense, those kind of things. So there's a peer review process. I have a couple publications in these, mostly anyway, so we'll go research reported, submitted to journal for review, and then the article is reviewed by two or more experts in the field. So these are the ones that you really want to look at for the real deal, so to speak. Okay, and we're not going to go into evaluating journals and all that. This is it's nice, but there's more rigorous ones and there's less. But for the most part, if it's a peer-reviewed journal, it's pretty legit, especially compared to all the internet stuff that's out there now. Okay, and there's more on those various journals. We don't have to know them. This one, Journal of Strength Conditioning, I used to get a lot because I was a CSCS, which stood for Certified Strength and Conditioning Specialist from the National Strength and Conditioning Association. Both of these I used to get a lot, but the International Journal of Sports Medicine I used to like quite a bit, and then the ACSM has the Medicine and Science and Sport and Exercise, and that's what I've been published mostly in my abstracts and stuff. They call that the MSSE, and again that's the ACSM, which is one of the more prestigious of the certifying bodies for personal trainers. Okay, and yeah, there's just more. Google Scholar is pretty good, so that's going to look at scientific literature. PubMed, of course, that's what the libraries used to always carry, and I'm not sure if you get that with at the school for free. I think you will. And then you do these various searches, so you do ors or nots or ands. Anyway, well, that's not part of our course. And this is the sections of a standard research report. So they're all written this way and they all must meet these specific requirements. It sounds boring and kind of canned. It's really not, though. This is just the way it's done, and it really helps keep things clear for those scientists that are even trying to recreate some of these studies. Okay. Yeah, they have critical evaluation, the research report. I'm not going to focus on this too much, but is the research question important? Is the research peer reviewed? Were the measures performed both valid and reliable? Okay, so those are the two things that we really want. And I'm just going to briefly tell you, if something is valid, that basically means you measured what you the way you measured something really measured that something. Okay, that kind of sounds confusing, but say I want to find out my height. I'm using a ruler and I'm actually finding out my height. Now that's clear and simple, but what about VO2? Does that relate to someone's performance in aerobic sports? Actually, it does because it says then reliable is if someone measured it, could another person measure it that same way and get a similar reading? So there's a lot of types of reliability as there's different types of validity, but that's basically the big difference between those two. Valid is you actually measured what you were trying to measure, and reliable is that measurement would stand up over time or different people given that same measurement technique. Studies well designed, that would be often with controls or even randomized controls. We'll get into that. And was the sample size sufficient? And the statistics will bear out on that because small sample sizes aren't good to try to get what they call statistical significance. And that would be just an example of how heart rate and exercise intensity and VO2 max in this case are really directly related. So yeah, if there's ever a very clean and clear relationship, it would be heart rate and say how fast you're running or how hard you're working out. Heart rate is very predictable on that. And there's uh, an article on that. So you can look at that on your own if you want. Okay, I'm not going to spend much time. These were the two I actually just mentioned. I've been a member of both of those, but there's a whole lot of other ones. This one's very reputable in Australia, and of course, the European College of Sports Science and British. So, but anyway, that's good if you're in those areas. Those are very good organizations. If you're going to personal training, I could talk to you more about that because I used to work for ACE, the American Council on Exercise, and that was my world for a while. And again, ACSM, personal trainer, group exercise, and they show in the National Strength Conditioning Association and the CSS. That's what it was for a long time. Okay, there's a lot of different areas going into exercise phys. I've been most of these actually at one time, even that one. But but we're going to keep going from here. And of course, professional schools, a lot of exercise phys people will go in these different directions. I was originally going to go into PT, but I had to do it over again. Yeah, PT or OT, kind of going that way. A lot of PAs are really common now to go through. So I educate a lot of people that want to become PAs. Okay, and again, more on the milestones. We're just going to pass by this. Okay, moving into chapter one. I'm going to go straight into this now. Um, this is the lecture outline. They're always going to give a lecture outline. So this would be good for you after you've gone through to see that you've hit these major points or understand these major points. Okay, so we're going to go pretty fast through here. Of course, you guys are all familiar with the metric system. It's actually based on water, which a lot of people don't realize, but and I'm not going to go into all the details on that one. But whenever you see 1 or 0 or something, you know it's based on water. But with that system, they have a System International and SI units, and there are certain units that are standardized for certain measurements. And we'll go into a couple of those. Generally, you won't have to memorize most of them. Knowing that watts is for power would probably be the one that I want you to know. And of course, it's all based on tens, so we're not going to go through it. But right there would be the sort of the cutoff um, for the hundredths versus the really small stuff here. But you've heard of nanobots or nanotechnology. Well, that's based on super small stuff here. And then, of course, above 1 would be these. And you could even go tera, and of course, all those. And again, these are the various SI units. Um, I may try to have you know a couple different ones, like, well, we're going to discuss these ones here. So force versus work versus energy versus power, all pretty closely related. Interestingly, work and energy are both joules. Newtons are based on joules, and then again, the power with the watts. And so velocity meters per second, Newton meters is for torque. So it's kind of an interesting one as well, the type of power. Okay, so let's go through some of these. I've done a couple additions to this, but work, okay, is force times distance. Go ahead and know that. So it's how much force can I exert across a certain distance. So if you are carrying, this is called a 40-pound box for 20 feet, you did a certain amount of work. And that was, again, force, I mean, the newtons was the force, the meters was the distance, and work is in joules. So now, what is a joule? Okay, it's 1 Newton meter is 1 joule. Okay, so that's kind of what they're showing. But it's also, it has different conversions. So 0.19, oh yeah, so that's the same with joules. Yeah, don't worry about these transitions right now. Okay. Moving on, we have work and energy. Okay, so kilocalories, you've heard of that one. It is a type of energy. If it's in food, it would be a stored energy, but it is energy. What is a calorie? Of course, it's the amount of energy to raise 1 gram or milliliter of water, 1 degree centigrade. Okay, so what's a kcal? It's the amount of energy to raise 1,000 grams or a kilogram, 1 degree centigrade. Whenever we are talking about food calories, we are talking about kilocalories. It's the stupidest thing in the world. I know. Why we can't just say kcal, or kilocalorie. No, but we have to just use the word calorie and then make it a capital C because we're too stupid to use the word kilocalorie. And excuse my harshness on that, but it's so crazy how people can't get that fact. Okay. Anyway, so joules is again it's a type of energy. Okay, so it's a term for energy and work, which is a but yeah, so you see how these are exchangeable. So the kcal and the joule can be related to each other. And also, well, we'll get into kpm, what that means is kilogram per meter, kilopound, as they're showing it. Okay, what is power? Okay, that's the amount of work, which is again force times distance divided by time. So it's how much work are you getting done in a certain amount of time. So the more power you have, the more work you'll get done in a given time. Okay, so that's important to know. And of course, if you had a lot more time and you could sustain a certain work level, you're going to have that power. But again, power is work divided by time. And again, this is in watts. That's the SI unit. Work is in joules. Of course, time is in seconds. and again, a watt. So whenever you hear about a kilowatt, when I used to cycle, we used to talk about how many watts could you maintain while you're cycling. Well, that meant both how fast you were turning your wheels and what resistance that wheel had on it. And of course, we used to use these old Monarch bikes that had, these belts that would be, having tension against them, and then you'd see how high you could go, and this little swing thing would show you. Now it's all digitized and stuff, but back in the old day, okay. So you've heard of a horsepower for an engine. That's also a term for power. So again, power is work per unit time. And yeah, so we'll just keep going from here. But kcals per minute would be a good example. Okay, how to calculate power? Yeah, they're going into it, but like I said, a watt is a joule second. Whenever they use this term, that means it's divided. It'd be the denominator. So basically, it's joule divided by a second. So it's the inverted thing. So that's always an inverted thing, meaning it's now the denominator. Yeah, so this many joules in that many seconds equal that many joules equals that many watts. Okay, so that's basically just dividing. And ergometry, okay, that's a study of work, basically, but it's a study of how much work output you had done. Um, for my eighth grade science project, my dad designed a finger ergometer. Okay, but again, the word ergometer measures the amount of work. So you have a cycle ergometer, you have bench step ergometer, you have arm ergometer. So all these different things can be used to measure work. And again, that's we'll keep going. Now, yeah, they're just showing the different ones. There's my old Monarch that I used to talk about, and like I said, you can see a little ball on there that would be swinging because of it, but now it's all digitized with fancy seats and all good stuff. And of course, treadmills, the faster you go on it. And then I did a lot of step tests on people, especially when I was doing corporal wellness. And let's not worry about this. Okay, using the bench step calculation of working power, we're not going to go into the details of this, but when you're measuring someone on those steps, you have to have them do a certain number of steps per minute. So you have a cadence then. And then however high the step is a certain amount of height. And then of course they're raising their entire body weight each time. So and they're saying in this case, a 70-kilogram person, basically 154 pounds, went 0.3 meters. Okay, so it's like a foot roughly, or you could say right around a foot, and then 30 steps per minute for 10 minutes. What was their total work? And it ends up being here that many joules or kilojoules, or that many Newton meters is kind of what the way of looking at it. I'm not going to have you do this kind of math. Some of you will probably say, "Whew," but I do want you to know what work is and the units that they're using to measure it. And again, power is work divided by time. Okay, and this was again, work is force times distance. So again, force times distance. I'm going to put this here divided by time. Okay, well, what is distance divided by time also known as? Velocity. What? Really? Yeah. So force times velocity is equal to the amount of power that something has. Again, that's measured in watts or joule seconds, which is a lot. What? No, watt. All right. And yeah, so we're just going to keep going on this as another example. And we're now looking at how to calculate it with a treadmill that's horizontal versus one vertical. Now, whenever you have an incline of slope, they're actually looking at how many times the belt moves. In this case, 100 units of belt movement. Okay, so that was but they're saying a 10% grade meant 10 meters rise for 100 meters of belt thing. It's basically rise over run. Okay, so this is 10 meters by 100 meters. I do hiking sometimes, and we're hiking 25, 30-degree things, but that meant I go 30 feet. I'm sorry, for every 100 feet that I'm traveling, when I'm doing those 30% grades, I'm going up 30 feet. And that's pretty damn steep. Okay, so anyway, that's just so you know, um, that'd be a steep grade. A lot of the grades people think of as steep are about 10%. That's so 30 is really steep. But anyway, the bottom line is that's what percent grade means is rise over run. And there's the same thing. We're not going to go into a lot of details on this. Just know for the case of the treadmill, you were going 10 meters up for every 100 meters that you were going out. But again, let's not worry about the trigonometry of it, the sign. And more on this. Um, we may even do a project where I'm going to have you calculate what your VO2 is for a certain walk compared to your predicted VO2 max. And then we'll look at your heart rate for the walk compared to your predicted heart rate max. So you can now relate what percent of max was my heart rate versus what percent of max was my VO2. That's a fun little experiment we're going to do down the road. Um, okay, work body, yeah. So again, this is just how you do it with a treadmill. Um, I'm not going to make you know the differences, but they're trying to look at vertical displacement or percent grade, and the distance shows you how much work you've done going up, and then you have to add how much work you've done horizontally as well. Okay, how do you measure energy expenditure? Through direct or indirect calorimetry. I actually could do indirect at my old lab, okay, when I had business for that. So I would measure the oxygen consumption using a mask where they actually had an O2 and CO2 sensor um, of the people, what they were breathing in and out. So it's kind of interesting. But we'll get into this in a bit. But just so you know, right now, I'm going to give a little triangle. METs is a unit that we'll be looking at, which stands for metabolic equivalent unit. But you can relate it to how many calories you're burning and how much O2 that you've had that you're consuming too. So the amount of O2 you're consuming is related to the amount of calories you burn, which is related to your MET level. And just kind of keep those things in mind. It's very an interesting way to, mostly this is what I look at. And we'll do a project looking at that a little later. And yep, so direct calorimetry is literally looking at foodstuffs and how much energy they put off. We actually did this in a chemistry once that I had where we burnt a Cheeto, and believe me, it burns big time. Okay, so and that's not even the hot Cheetos, but the Cheetos were incredibly big flame, and then we'd actually have to measure the ashes because it wasn't fully combusted, obviously, and but that's how many calories it was burning, not kilocalories. Okay, so when they say 1,000 calories, that's only one kcal, which is only one what we call true calorie, in a food. So needless to say, our food has a lot of energy in it from a realistic standpoint. And let's not worry about the details on how they calculate this. And let's not worry about the details on how they calculate this. But again, back to the indirect, okay, is using somehow a treadmill or whatever, but they're measuring the oxygen, the VO2 that you're consuming. Okay, so we'll get into that in a bit, but it is based on the amount of oxygen you consume. We can find out the number of calories that you expended. So they're showing here the example of VO2 is 2 liters per minute was approximately 10 kcals or 42 kilojoules per minute. And that's how much the person will burn. And this is how you can measure it. So again, with O2 and CO2 analyzers, I had one called New Leaf. It was, but they have better ones now in various labs. And let's see, want to go into this yet? No, no, we don't need to go in. But when you're trying to do an exercise test on someone, you're going to try to calculate their what they call VO2 max. So you predict how much work they're doing versus how much oxygen they're using. And then somewhere in there, you would predict if they did a maximum amount of work, what their VO2 max was. Or you can do it on a ramp test too and assume that certain things. But anyway, but back to this is uh, it is based on the amount of oxygen you're actually consuming. And more on this. Yeah. So you can again calculate how many calories you're burning based on how many liters of oxygen you're consuming, again, in this case, per minute. Interestingly, just to let you know, and this is a total side thing, but um, inspired air, I should say, is roughly 20.9, and I say roughly, and I'm being quite accurate. But inspired air is about that. So let's just say someone's breathing in roughly 20.9%. And then we measure what they're breathing out. Okay, we'll call this out. And say it went down to 13.9. We'll just do this for easier numbers. We know that they consumed 7 and then whatever the units is. In this case, it could be liters per minute or whatever it would be was their consumption. So you're actually measuring what's it. We know what's incoming, but we measure what's outgoing, and then we take that difference to figure out what that was consumed by the body. And this is 1 MET resting. So I'm going to go ahead and have you know this. When you're resting right now, if you're sitting down doing nothing, basically not even not talking, you'd be at 1 MET, okay, which is 3.5 mLs of oxygen per kilogram per minute. Now, what's also interesting about the METs is they're directly related to your caloric expenditure per hour per kilogram. Okay. So say I weigh, I'm pretty close to 80, actually a touch over 80 kg now. Okay. And say in a 24-hour period, I wanted to find out how many calories I burned doing nothing, just at 1 MET, resting. And so what is that? 32 and 160 is 1 6. Oops, 1 9. See, now I'm getting messed up. See, 32 and 160. Yeah, so 1920. And that's probably, yep, pretty close to what my RMR would be. Is that right around there? And I'm not sure I even did the math right. But anyway, let's keep going. And they're showing energy expenditure can be estimated during many types of physical activity, basically anywhere where you can measure oxygen, but it's commonly done running, walking, and stationary cycling. Could you do it on a swimmer? Yeah, they actually can do it on swimming as well. Horse riding, anything where, and they actually have some pretty interesting mobile units now that the person can have while they're traveling somewhere. That's it. And again, we'll be discussing what VO2 means, but kind of know the units right now. Again, milliliters of oxygen per kilogram per minute. So somebody's size matters as far as their absolute, but if you look at it per amount of weight that they have, that's how it's related. And yeah, 50 or 60 would be pretty high. Um, I've had mine actually measured at 70 at one time. That was probably my top shape ever. But yeah, they're looking at walking and running speeds, and this is meters per minute. We're actually going to calculate, like I said, ours, and it's using this exact formula. So we're going to find out how many meters per minute we walk, and then that'll be multiplied by 0.1. Then we're going to add 3.5. Most people land in the low teens area, so 13, 15, something like that. Okay, and now they're looking at estimation of treadmill walking versus treadmill running, and there are different formulas for that. But we'll show you a bit of both. But again, we're going to do one on you guys walking compared to, again, the Rockport walk test VO2 max estimation. And again, remember that the heart rate was very related to the VO2. Well, the VO2 is very related to the amount of work that you're producing. Thus, heart rate would be very related to the amount of work you're producing. So guess what? When you run faster, your heart rate goes faster. Wow. What if you cycle faster? If you run up a hill, you're doing so yeah, it's kind of interesting how actually in the world of cycling, they're really all about the work rate or watts, and they actually can calculate that on the bike. So they can estimate the amount of actual work you're doing. So if you're going up a hill versus high wind resistance versus all these things. Anyway, it's kind of complex, but they look at pedal pressure, believe it or not. So, all right. And again, this is how a lot of it's done. I actually met one of the guys that was total specialist in that. I'm trying to remember his name right now. He died very early on, very early. I'm not sure exactly the reason, but yeah, he was the god of cycling and ergonomics. Okay, we're going to get into a couple more terms here. Efficiency, and we're going to also look at economy down the road, but the ratio of work output to energy input. Okay, so what it's basically saying is how much work are we getting out of something for every amount that we put in. In other words, what's not wasted, or I should say, what's wasted is the difference between these two. So the more efficient an individual uses less energy to perform the same amount of work. Now, does that mean if they were not going in a straight line, they're still producing that amount of work output, but they're not that's an economy thing. Well, we'll get into that in a second here, but it's basically saying for all the energy I put into something, what's coming out of it? And remember how the light bulbs, there's a good example right now. How many lumens, how much light are you getting for a given amount of energy put in a light bulb? Well, we all know that the LEDs are crazy more efficient than the old light bulbs were, where they were producing a lot of heat. Yeah, they were giving off some light, but they were wasting a lot of that energy into heat versus doing the light. Okay. And then we'll look at net efficiency too, what that means. Okay, and here's what percent net efficiency is. So again, it's the work you got out of something versus the energy you had to put in above rest. Again, above rest. That's what the net part of it is. Okay. So the net efficiency of cycle ergometry ranges between 15 to 27. That doesn't sound very good for our human body, though. That's not bad. Okay. So and I don't know if that's trained cyclists or not. Probably the 27 is versus the 15 is not. And here they're showing about 22% for this particular person. So, how many calories went into it, okay, versus what work did they get out of it? Okay, so that would be kind of and we're not going to go into the details on this, but they're basically showing how they had 1.4 kcal per minute into it, and then the net energy expenditure, what they got out of it. And so as a result, it was only 22% efficient, 22.6. Okay, what is exercise intensity? That can be measured a bunch of different ways, okay? But the bottom line is its relationship between work rate and energy expenditure. Okay, so again, we'll look at it several different ways. Um, Efficiency decreases at high exercise intensities. Go ahead and know that. So the harder you have to go, it's kind of like a car too. You took a car really high speeds, okay, you're going to be using more gas per that speed than you would at a lower speed. Okay, so the higher exercise intensities so-called waste more energy in other ways than the lower ones. High cost of heavy exercise. So you're trying to move something real fast. So the speed of movement has a lot to do with this and the muscle fiber type. So slower muscle fibers, type 1, are called slow oxidative. They're much more efficient than the fast. And we'll get into some of the bioenergetics of it and all. But generally, again, the slower you could go, the less heavy engine. You could use a very efficient, fuel efficient engine versus if you have to go fast, you have to rely on uh, let's just call it a different kind of engine or a gas that would take up a lot more energy, less efficient. We're going into the details here. I'm not going to spend too much time here. They're showing the arms are pretty efficient though, compared to what is this? Just other things. So as you can see, after a certain point, um, the work rate is is decreased compared to the energy expenditure. So there's a less efficiency at higher work rates. And again, same thing here. Interestingly, though, there's an optimum. Okay, so somewhere in there, this is whenever you look at what they call inverted U curves, know that they're speaking of an optimum, meaning too little or too much is not going to hit your peak. So in this case, we'll go with the 75, the pedal rate. And yep, that's about what they've shown with the really well trained the Tour de France type guys can rev pretty fast quite efficiently, but generally 60, 80. Same with the step rate, believe it or not. We found around, I think it's double that. Well, 180. I'll have to look back at those, but how quickly you step, there's a most efficient step rate or cadence. Oh, here it is with running economy and but lower VO2s, same speed indicates better running economy. So, would you want to be spending less oxygen for a given speed? Now, was this the same as efficiency? No. And don't worry about the running part of it. Just look at economy itself. So, we want to talk about cost of running at a certain speed. So now we're kind of getting the cost. That's the economy part of it. So slow muscle fibers are more efficient at using ATP, individual differences in running economy. So elite runners are more efficient compared to non-elite. That means that they can run with a lower economy for a given speed. And there's a difference between elite runners and untrained runners. It's a 10% difference, as they're saying. So they can spend less oxygen for a given speed um, than not untrained runner. And that was the end of that chapter. Kind of heavy duty, I know. Okay, getting into a final chapter 2 of this presentation. Now we're going to be looking at several things regarding the body, but the main thing is called homeostasis. That is the driving theme of our entire body, and what it's all trying to strive for is maintaining homeostasis. And we'll get into that. The bottom line is it's trying to find a balance point for whatever system um, and whatever that system is going through. We'll explain to you more. Okay, so what is the example of homeostasis? Maintenance of a constant, okay, and a normal internal environment. Now, they shouldn't have said constant because it's not being held steady. It's just being held within a certain parameter. Okay, but heart rate, body temperature, arterial blood pressure, all these things are held at a relatively normal thing, despite what the external environment is putting onto us. We will try to maintain a certain body temperature, heart rate, and blood pressure, no matter what. Okay. And we do it because we have corrective mechanisms in our body to either raise something or lower it. But important to know, if it was too low, our body will try to raise it. So it's always opposite. The stimulus and the reaction are always opposite with this type of mechanism, which is known as negative feedback. Okay, Claude Bernard, very famous physiologist. And yes, okay, so what is a steady state? And this isn't important to know. The variable is unchanging, but like I said, not necessarily normal resting value. So if you were running, you obtain some higher heart rate, but eventually you'd stay at that heart rate for that running speed. Say you run a little faster, you'd have a little higher heart rate. It takes about 3 minutes roughly for someone to maintain a true steady state. You can get there a little bit faster, say two. And of course, the bigger the difference between resting and this new level, the longer it would take for you to get to that steady state. But that's what's interesting about the body. So it can have a higher heart rate, but it'll keep that heart rate for that given running speed. Or the body temperature will raise, but try to be maintained for that particular condition. And so here they're showing core temperature. Yep. And so it got raised, but then eventually it hit normal. So 37 is generally our normal body temperature. In this case, whatever they were doing, it stabilized at just under 38. Okay. And yeah, intracellular control systems include these, and organ systems have these. Okay. So protein breakdown and synthesis, energy production, maintenance of stored nutrients. Those are things that the intracellular control systems do. Don't worry about these right now. I'm not concerned. Okay, I am concerned you know these big three though. Okay, this is a classic biological control system, and it's got 1, 2, 3 parts to it. Okay, and the receptor or sensor is always what detects whatever's out there. It's going to go to the control center, or sometimes called integration center. What would that be most of the time? The brain. The brain would be our best integration center. So it's taking in the information and then pushing it out to the effector that can do something about it. So say you were trying to lift a weight and the receptor found out, wow, this is heavy. Okay, so it told you it was heavy. Your brain said, I'm going to need to recruit more muscle for this. And then it sent out the nerve impulse to hit bigger muscles and more of them, to more motor units to then be able to lift that weight. So that would be a class example. And then the effector said back to the sensor, no, it doesn't. The effector then does what it needs to do, and your body feels, oh, I succeeded. But that is a classic, again, control system, or what we call feedback loop. And here it is again. So again, the sensor, the control center or integration center, often called, and then the effector. It's often a gland or a muscle. And back to homeostasis. Okay, they're going to go through more on this, but again, negative feedback is trying to pull things toward a set point or toward the middle. So if something's too high, it pulls it back down. If something's too low, it pulls it back up. So that's just the way negative feedback works. Most systems in our body work on this mechanism. Interestingly, there's something called oxytocin, which works in both the milk letdown for a baby and for breastfeeding and for labor. So the two things kind of similar. So the uterine contractions, but that's because you want those things to be over with. So that's going to actually drag things to the extreme. So the stimulus becomes the positive to have more of that stimulus in there. So anyway, we'll get into this in a second too. But most feedback is negative feedback, trying to control things. And here they're going through another one. But oh yeah, so this is where it's positive feedback, and again, very few. So they're using the one I just said, oxytocin, from the pituitary gland, and again, basically, it's the baby's head pushes on the uterus, which then causes oxytocin to get released, where the brain realizes it, releases oxytocin from the posterior pituitary gland, which then causes more uterine contraction, which then causes more pressure of the baby's head, which causes more contraction, blah, blah, blah. So, okay. A gain of a control system. That's a good thing to know. It's basically the accuracy of it. So a system with a large gain is more capable of maintaining homeostasis than with a low gain. It's basically saying how sensitive it is. So a larger gain is greater sensitivity, and they have more ability to adapt with that. So they can gain more or gain less. Okay, so that's a good way of putting it. And yeah, some examples of this, we regulate body temperature, we regulate blood glucose. Both of these have to be maintained within certain ranges, or we're no longer in homeostasis, and we have real trouble with that, and we may even die. So elevated blood glucose signals to the pancreas, causing insulin to be released to lower it. So always kind of keep that in mind right now too. Insulin lowers blood glucose while glucagon raises it. But it's a combination of those two that maintain it. And it's right around 90 to 100 milligrams per deciliter, what we call percent. So anyway, that's what the normal blood resting, fasting blood glucose should be. And here they're going into the different control parts, but this is how it affects it one way or the other. In this case, it was for body temperature. Yeah. So we can sweat or we can shiver, but either way, we're going to try to maintain 37-degree body temperature regardless. And they're going into this a bit more. So exercise disrupts homeostasis by producing changes in these big three. Get into these later on, but that's very important to know. These big three and temperature, thermal, are all detected by the body and trying to maintain it. So if you were exercising, would you become more acidic? Yes, that's actually a lower pH. Would you be lower in the PO2, the amount of oxygen you have? Yes. Would you have higher amounts of carbon dioxide? Yes. Well, our body will react to try to balance these. And it says during submaximal in a cool environment versus intense exercise in a hot environment. The end result is fatigue and cessation of exercise. So you were able to maintain it here, but you weren't able to maintain it here. And they're going into the situation with diabetes. We're going to just go ahead and pass by this one right now, especially with type 1. And more on that. So insulin is no longer produced. This is why they need insulin to control our blood glucose. Okay, we're coming toward the end here, but I want to go through the two different A's. Adaptation versus acclimation. Okay, so these are both trying to get used to something, but what's an adaptation? It's actually a change in structure and function, of a cell or organ system, results in improved ability to maintain homeostasis. Many adaptive changes occur within cells. What is one of the main stimuli we're going to be looking at to find these adaptations? Yes, exercise. Okay. Acclimation, adaptation to environmental stresses, for example, results in improved function of existing homeostatic mechanisms. Interesting here. If you were in a hot human environment a lot, you'd sweat a lot. Eventually, the body would start secreting a more dilute sweat, but actually start sweating earlier in when you got hot because it literally got used to it. anyway, so we, of course, plants and animals do this all the time to adapt to their environments. They're adapting, but at first they have to, and of course, when you're, say, at altitude, you have to acclimatize toward that. So you're changing things, but temporarily. But then eventually, the structure and functions change. And cell signaling is one of the ways that this happens. And so we'll get into this more, but chemical um, signals are sent. They're known as cytokines. Cytokines are chemicals that signal one or have one cell signal to another. And again, very important for maintaining homeostasis. Oh, hormesis. This is a good, interesting too. So go ahead and know this term. Processes in which a low to moderate dose of a potentially harmful stress, for example, caging, results in a beneficial adaptive response on the cell or organ system. Okay, so it's basically how you tolerate something in, in quantities. So exercise-induced hormesis defines much of what we know about exercise-induced adaptations. So if you got tired from running, say, 5 miles, but you kept doing it, eventually you would adapt to something. Was that a harmful stress? Yeah, maybe, but the bottom line is your body interpreted it that way, and then it made adaptations to make sure it can handle it down the road. But go ahead and know that term. Hormesis was the term for this adaptation. And here they're just showing. So too little exercise or too much is not optimal. You want to find this optimal amount so that your body can adapt correctly. And again, this has to be done at a certain pace depending on that person. So they can either, um, if you, like I said, overtrained, you're gonna, your body's going to be maladapted. It's going to go, it's not going to find the time. And of course, what causes adaptation? Nutrition and sleep, are two of the main things that allow for this adaptation to take place. But the insult has to happen in there too. They call it an insult, the exercise. But anyway, this is a good graph here, and more on that in this article. Okay, cell signaling pathways promote cellular adaptation. Cell signaling. What we call those things again? Cytokines. Communication between cells with chemical messengers. Again, it's maintaining homeostasis, and the cell signaling pathways exist in cells. We're learning a lot about these different mechanisms, even between, say, the brain and the immune system, between the gut and the brain and the immune system, and all that kind of things. And these are different ones. Go ahead and know one or two of these. I'm going to go ahead and show you a couple. Juxta means next to. Think next to. So pass between two connected cells next to it. Autocrine is within the same cell. So auto is same. And then paracrine is nearby. So go ahead and know this. So it's next to it, same cell, nearby. Endocrine, of course, can be far away. So it can be from something quite far away. But again, it's released into the blood with a hormone. Those are three kinds of crins, four kinds of crins. And don't worry about these extracellular vesicles so much. They're the way they communicate, say, with the endocrine or with neurotransmitters. But this is the way that things communicate as well. And here they're showing the different mechanisms. Um, Integrin, again, chemical messenger triggers a pathway. Here it's next to the cell next to it. Here it's in the same cell. Here it's nearby cells, and here it can be pretty far away. But again, had to go into blood and had to have a hormone receptor complex. That's what's unique about endocrine. And then finally, they're just showing here how endocrines affect it. So exercise-induced release of these different vesicles can travel to these different parts to cause some kind of an adaptation. So white fat cells become brown or beige, as they call it. Skeletal muscle adapts in positive ways to withstand greater exercise. Epithelial cells do. The brain and muscle talk to each other. So this is the way they talk to each other. And yeah, stress proteins, heat shock proteins. Interesting, there was a program with Chris Hemsworth that was where Thor, good looking Thor, was going through these kind of hard situations. But one of them they showed where he was going through freezing water. He had to swim a certain amount of time in the absolutely frozen water. But they were showing another time where he was in a sauna and the heat shock proteins could help repair damaged proteins and cells. So some athletes are starting to do this hot and cold treatments to induce some of this. And like I said, stress that induce stress proteins include these. Okay. And some of these are good or bad, but high intensity or prolonged exercise can promote these stresses as well. So anyway, that's how some people are getting conditioning. It's kind of a cutting edge stuff right now, especially with the high temperature stuff. And more on the cell signaling. We're not going to go into all these details, but we do get exercise induced protein synthesis, aka hypertrophy, because of the talking of this, but we actually call transcriptional activators will then bind. A lot of this is happening through what they call satellite cells. So it's really fascinating stuff. But exercises induces these signaling things to activate transcriptors. That's what steroid hormones do as well. They actually go straight into the nucleus to cause transcriptional activation to actually make more of a certain protein to deliver a certain function. So again, resistance and endurance exercise promotes activities, so they promote the different cell signaling activities. And here we go. Are we looking at satellite cells? Not yet. Yeah, but pretty close to it. So they're just showing here in a muscle how this works. Again, we don't need to know the details on this, but it is interesting how, again, exercise can cause some sort of cell signaling to actually cause the transcriptional process to kick in this case, an mRNA for who knows what, maybe more muscle. And there's your article. And that's the end of it. So remember there was something on dynamic consistency or constancy of homeostasis. What was the biological control systems in our body, the nature of those control systems, positive, negative feedback, examples of a homeostatic control, the test of a homeostatic control. So that was the moderate exercise in a cool environment versus high intensity exercise in a hot environment. Exercise improves the homeostatic control via cellular adaptation. What was that called? Hormesis, right? And then stress-induced proteins assist in the cellular regulation of cellular homeostasis. And again, some of that's for stuff we see during resistance training, and like I said, with satellite cells and with mTORs and all these really fancy things that are at the cellular level. All right, have a good one. That was a good chat. Our first one of what is it, 11 at least we're going to have of these.